Hello Brain Shakers, welcome to the Brain Shakers Academy. Your host here, Brave Alistair. In today's session, we're going to be looking at tissues. Now, tissues are just a group of similar cells with their extracellular matrix that are then put together to perform a similar function. Now, the study of these tissues is a process that we call histology, meaning you have to get into the nitty-gritties to have a clear understanding of what these tissues look like and what functions they then perform. Now, we are going to dive into today's session to basically look at these different tissues. So the body has about 4 principal tissues in it, and I have drawn a diagram here that is going to help us to understand what tissues are and how they relate one cell to another. But before we even get to the diagram here, we need to look at some of those tissues that the body has. We said that they have 4 primary or principal tissues. And number 1, we can talk about the epithelial tissue. So we have an epithelial tissue, we have a muscle tissue, we have connective tissues, and then we also have a nervous tissue. Okay, so those are the 4 principal tissues that we're going to be looking at. Now, if we take out the 3 and 4, this particular session just delve into epithelial tissues and understand what these epithelial tissues are. Now, the epithelial tissues can actually be broken down into 2 forms. That is covering epithelia, and then we also have what we refer to as glandular epithelia. Now, for this session, we'll be looking at covering epithelia because I look at glandular epithelia in another separate video so that we can look at its entire nomenclature and look at what the epithelial tissue is going to basically be doing when it is lining those glands. So the main function of these epithelial tissues is to basically line body cavities or cover body surfaces. Now, let's look at the build up or how these epithelial tissues are actually formed. So the epithelial tissue is comprised of cells and also has a connective tissue on the background which creates its basement membrane or the extracellular matrix. If you look at this diagram that we have here, you are going to notice that the ones that you see in blue here is epithelial cell here, and then you have at the bottom end here, you have a connective tissue coming into play. Now, when we are looking at this connective tissue, you also see that this is the capillary. So you have a capillary at the base here because these cells, they will also need some element of nutrition, and so they have to get the nutrition from the circulatory system, and they may be innervated to supply the stimulation for them to then perform that desired function. So you also find that you have nerves here. So this is a nerve ending here that you can see it is actually getting access to the cells themselves. Before we get to the cell, just on top of this lamina propria, you will notice that there is this particular layer here. Now, this particular layer is what we refer to as the basement membrane. So this is the basement membrane. The basement membrane is an important structure because it houses 2 important layers. At the bottom end, you have fibers here, and these fibers, we call them reticular fibers. So these reticular fibers, they form the basement of that basement membrane, and then we call it a reticular lamina. Then the upper part here, which is going to have the base of the cells, where the cells actually sit, is what we will refer to as the basolamina. Now, basal lamina is being produced or secreted by the epithelial cells, whereas the reticular fibers or the reticular lamina is being secreted by the connective tissue down there, and then you will have your epithelial cells there. So meaning the epithelial cells will always sit on a basement membrane, meaning they have somewhere to actually hinge on, so that as they perform their function, then they are formidable in their structure as well. As we have looked at the buildup of these cells, we also need to understand that there are intercellular adhesions, or things that actually hold these cells together. Now, when we are looking at these intercellular adhesions, I want to make mention that there are a number of adhesions that we can talk about. If we look at these adhesions, the black markings here that actually hold the cells closer one to another, these are the ones that we are going to call tight junctions. These play a key role in controlling the movement of substances from the cell, going out of the cell, and from out of the cell into the cell. So they form that tight junction between cell to cell, controlling the movement of solutes there. From the tight junctions here, you will have a belt here of intermediate filaments, and these are going to be intermediate junctions. So we have what we refer to as intermediate junctions. So these are also known as adherent. Okay, adherent junctions. It forms more or less like a belt to hold the cells together. And as it is holding these cells together, what is then going to happen now is that even if there is any pressure that is applied on the cells will have support from each other because of that belt that is actually well formed around that region. And then you have this portion here that is going to be a little bit open, and you have what we call the linker glycoprotein. These linker glycoproteins, they are in the form of cadherin. Okay, so cadherin is just a glycoprotein that is just going to help the cell attach one to another. And this whole complex now, the whole complex that is then formed is what we are going to call desmosomes. From the desmosomes, you also have what is known as gap junctions, which are more in the form of connexons, connecting cell to cell. These ones here are what we are referring to as the gap junctions. Now, gap junctions are also very important in the transmission of the nerve impulses or the action potential, because they facilitate faster movement or a quicker activation of that action potential from one cell to another cell, so that when the cells have to perform their function, they then perform the function at the same time, because they'll be stimulated at the same time by the facilitation of that movement of the action potential by the gap junctions here. Now, at the bottom end here, you have another type of connexon that is going to be connecting the cell to its basement membrane. Now, what is connecting the cell to its basement membrane here is what we are going to refer to as a hemidesmosome. Now, it's a hemidesmosome because there's no aspect of that desmosome from the basal lamina. It is only coming from the epithelial cell to actually attach itself to the basement membrane. Now, that is holding the cell and giving it its base. You notice that there is a blister, then what is affected is the hemidesmosome. There are other autoimmune diseases such as pemphigus vulgaris, where the antibodies that are produced during that autoimmune disease are going to come and then bind to the protein element of the hemidesmosomes there, preventing it from then attaching itself to the basal lamina, and then you notice so much of that blistering. How do we then classify the epithelial tissue? So we can classify epithelial tissues in three forms. Number one, according to the number of layers. So how many layers does that tissue have? Number two, according to the shape. So we're looking at the shape of the superficial layer or the topmost layer. So it doesn't matter if the bottom layer is holding a cuboidal cell, then on top we have the squamous type of cell, then we will follow the shape of the topmost layer. Number 3, we will basically be looking at the presence of specializations. Here we'll be talking about issues to do with the microvilli. We'll be talking to do with things to do with the presence of cilia as well. Now, as you can see on this one, you have this part here, which is a presence of specialization. So that is another way we can be classifying these epithelial tissues. Now, let's quickly look at some of those examples that we can cite about epithelial tissues. Number 1, you have what we would then be calling a simple epithelial tissues. So simple epithelial tissues, this is where you have just a layer of an epithelial tissue. So it could be a squamous. So number 1, we could be talking of a simple squamous epithelium. So that means that the cells are just more or less like fish scales. Okay, so you have that, and then it has a basement membrane also on the bottom part. That is a simple squamous cell. Number 2, you can talk of a simple cuboidal. So you can talk of simple cuboidal cells. The cells are going to be more or less like squares. So you have those cells. That is going to be considered as a simple cuboidal. It has a single layer of cuboidal cells. Number 3, you will have a simple columnar epithelium. So simple columnar epithelium, they are going to be more long and rectangular in shape, and then you have your basement membrane there. Those you are going to call as a simple columnar epithelium. You may also have what is referred to as a pseudo stratified epithelium. Now, to be stratified is basically to be put or placed in layers. So when we are looking at the classification of these epithelial tissues, it's either we have a simple epithelium or we have a stratified epithelium. Now, there are times when you have an epithelium that appears to be stratified when in actual sense it is not stratified. So what that simply means is that if you have your basement membrane here and then you have cells here, then you also have cells that touch the basement and some cells that do not reach the apical aspect, and you have cells on this other end here that do not reach the basement. So you have cells in that nature. So it appears as if it is placed in layers, but some of the cells here that you have, such as this cell, it does not reach the basement membrane here, and you cannot actually say that this is another layer because it is not well defined to be in layers. So this may be referred to as a pseudo-stratified layer. So in terms of tissues, you can have also what we are calling the pseudo-stratified columnar epithelium. Now, if you have a pseudo-stratified columnar epithelium, usually it is going to have the presence of cilia on it. So we'll call it the pseudo-stratified columnar ciliated epithelium. And this is what you're going to find in the respiratory tract, such as the nostrils. You will find a pseudo-stratified columnar ciliated epithelium. You will also have a simple columnar ciliated. So if you have a simple columnar ciliated epithelium, so this one is going to be found mostly in the oviduct. You find this one in the female reproductive system. Because of that cilia, it has to actually propel the ovum closer to the place of implantation or to the place of fertilization, and then continue to propel it to the place of implantation. So that is your simple columnar ciliated epithelium. Now, for epithelium that is stratified, in most cases you're going to have what is referred to as a stratified squamous epithelium. Now, you must also note that this is stratified. So it means that we are following the second method of classifying epithelial tissues, where we are classifying the epithelial tissue based on the superficial layer of the tissue that we're looking at. So if we have a basement membrane here, and then we have cuboidal cells here, we have all these cuboidal cells, and then on top we have the scales, which we are calling the squamous cells. So this, we will then utilize the topmost layer and call it a stratified squamous epithelium, without paying key attention to the bottom layer. So the stratified squamous epithelium, you will find it mostly in areas where there is going to be consistent mechanical and physical abrasion, such as in the oral cavity. You find it in the pharynx, you find it in the esophagus, you find it in the cervix, in the anal area, and also in the vagina. Any area that has more mechanical and physical abrasion, you will find a stratified squamous epithelium. Now, from stratified squamous epithelium, you also have another type that we can also call the transitional. So transitional epithelia, that is also classified under stratified. It is also called urothelia because you only find it in the bladder. Now, the urothelia or the transitional epithelium, when you're looking at the bladder in a non-distended state, it will appear to have about 4 to 5 layers. But when it is in its distended state, it will appear to have about 2 to 3 layers. Now, when you are looking at the simple epithelium, such as the simple squamous, that's the type of epithelium that you're going to find lining or structures such as the lungs and the blood vessels to allow for easy gaseous exchange. So cuboidal is going to line glands such as the pancreas and all these other glands. The simple columnar, you will find it in the smaller intestines. This one is not ciliated. So columnar, you find it in the smaller intestines where you are going to find the microvilli there. If you have a simple columnar ciliated, then that one changes and you will find that one in the oviduct. For clinical application processes, you notice that any type of tumors that are emanating from the epithelial cells themselves, in most common cases, they will be benign tumors. And if it is coming from the entire epithelial tissue or from the epithelia, these ones are going to be referred to as carcinomas. And if they are coming from the glandular epithelia, which we are obviously going to look at in a separate video, then those ones are going to be called adenocarcinomas. Okay, so basically that is the epithelial tissue. So if you did find this particular video helpful in understanding what an epithelial tissue is and how that epithelial tissue plays its key role, don't hesitate to drop me comments in the comment section and share the video as much as possible. Head on to the YouTube channel and subscribe, hit the notification button so you don't miss any of that. And as always, thank you so much for watching, and I will see you in the next one.